So I'm here today to tell you a bit more about what we do on the XChem platform in terms of fragment screening. So first, I'll just summarize a bit the two strategies that are being followed in drug discovery, mostly by industrial users. So there are two types of screening. You have the high throughput screening here on the left-hand side, where you will be looking for potent molecules, which are usually complex molecules and pretty big. So the probability to find such a molecule is low. That means you need to screen quite a huge amount of complex molecules. And then there is the approach of fragment screening, where you basically deconvolute those big molecules into small fragments. And the probability to find those fragments are much higher, meaning that you do not need to screen as many compounds as for the high throughput screening method. You probably need about 1,000 small molecules, but those fragments will have a weak affinity to your protein of interest, meaning you need to have a very sensitive technique to detect it. And you will achieve potency for those fragments through development of those chemical elaboration of those particles. And so why do we want to do that in X-ray crystallography? Because X-ray crystallography is a sensitive enhanced detection method. It can detect things in the millimolar range. It provides you then what is extremely crucial for developing those fragments into lead compounds, it gives you structural information. You know where it interacts and how it interacts, and hopefully you know where you can go. Then recently, basically it could be done in X-ray crystallography because of the throughput of producing those data. Now synchrotron can collect data in less than a few seconds. So data comes through very quickly, and the bottleneck was the sample preparation. You needed an infrastructure for people to be able to streamline that process. And that's what XChem has been put up together for. Does it work? So I've seen in the Practical Fragments blogs that there were 2 drug compounds in the market coming out from fragments, and there are about 40 compounds in clinical trials. So it seems to work somehow. In terms of the XChem platform, it was part of Diamond's user program about 5 years ago. It really started kicking in when they gave us dedicated beam time in 2015. So then between 2015 to 2017, we went from 5 projects to regularly taking more than 30, and especially the industrial users started coming in. And also we could take some in-house projects. At the end of 2017 and start of 2018, then we went really fully in the Diamond's peer review process with 2 calls per year. And also in 2018, Diamond really invested in our platform in the sense that it allowed the industrial to double their capacity in the lab. And also we created new access mode for the users. And in 2019, this is when we took the first back users. So for the academic users, the XChem is part of the peer review process. We have 2 calls per year in April and in October, and for the 2 allocation periods of 6 months. So if your proposal has been accepted during those things, it needs to happen within those 6 months at the allocation period. We have 3 access modes, the tier 1, which is basically an exploratory project. You just want to prove that the target is druggable, more or less, and you might not have necessarily lined up already all your collaborations for the ComChem and the biophysics. So it's basically a step for proof of concept. What you get though is 1 shift of beam time, which is about 8 hours. And then the tier 2 access mode, those are for the established project. So basically, you already know that this target is interesting, you already have the medchem lineup, the biophysics lineup, and you want really to do the full screen. So for that, we give you 4 shifts of beam time, and that should be enough to cover all the needs. Then recently, as I said, we started the block allocation group, which is similar to the bag for the MX beam time. You get a number of shifts defined for 2 years. and they are defined based on the number of projects you've put in the back proposal. But the biggest difference is that you have a super user in place, and those super users are basically expert XChem user who is going to coordinate and prioritize all the projects within the bag, and also train some of their users, some of their bag members for using the XChem platform. Then for the industrial, you can basically contact Alex or Elsa, and they have two modes of services. You come and you do your experiments, or they do it for you. All the information is in that link here. So what do you get when your proposal is accepted? Basically, you get the infrastructure of the XChem lab to allow you to prepare the samples. You come with your crystallization plates, or you ship us the proteins and all what we need to do the crystallization for you. You can also ship us the fragment libraries if you're not interested in the ones that we have. It just needs to be in the format that is compatible with the eco dispensing liquid that we have. Then you prepare all the samples. It goes onto the beamline, the IO41 beamline, where everything will be collected remotely and unattended, so you don't need to be on site. And hopefully what you're getting out of this exercise is a full mapping of potential interactions of your protein between your protein and your ligand in that crystal system. So it should give you a full picture of what is available in terms of interactions. We do 1 compound per crystal. We can handle about 1,000 to 1,500 crystals per week. And we also have then the data analysis pipeline downstream for you to basically detect those hits. So in a little bit more detail, the XChem workflow is summarized here. So on the top part is what is happening in the lab, and at the bottom part is what is happening downstream for data analysis. So you basically have the libraries that have a code that is being tracked down within SoakDB through all the experiments that you have to do until it gets collected. So the different steps in the lab, we have the crystal targeting, where you will need to decide which drops to use for soaking and where you want the soaking to happen, where you want to dispense your compound to. We do the dispensing using the ecosystem, which is an acoustic wave dispensing liquid dispensing system. It's very accurate and it provides very small volume. And then we have the shifter for the harvesting of the crystals, which is an XY stage. And it has a software to basically, again, track down what you're mounting and where it goes into the puck. Then all that information is, of course, barcoded and being tracked down, and is passed on onto the iSpy-B system for sample registration for data collection. And that iSpy-B is on all the synchrotrons, European synchrotrons, as far as I know. So then the crystals get collected. All the auto-processing results from that data collection is passed onto the XChem Explorer, which again talk to the same SQLite database and SoakDB, so that you're now tracking down the compound to the data set. And also XChem Explorer managed all the amount of data that you're creating and launches Panda software, which is a software for detecting weak binders in electron density maps, and also has a plugin for CRUD to do model refinement. So at the end, you have the full pipeline to hopefully go from the crystal to the structure with all your ligands down. So in practical, what we have a lab that has 2 ecosystems for dispensing your compounds and 3 shifter stations for your harvesting, crystal harvesting. We also have a dedicated team. Darren is focused on the academic with me, and Elsa and Alex are dealing with the industrial. Realistically, we can take 3 groups in total in that lab. So usually we have 2 groups of academic people and 1 group of industrial user. And this is how the lab could look like. That was the last training before the lockdown. So it can get really crowded. And as I said, we have dedicated beam time. So they are dedicated, they are marked in blue here. And we tend to use most of the beam time that we have in orange here. So there are 24 hours for the academic users, 16 hours for the industrial, and 8 hours for in-house research. And those are all XChem beam time. Right, so what we provide on top of that is an extensive user support. So when your proposal gets accepted, basically you'll probably get a phone call from us to figure out a bit. What we need to know from you is your crystal system. And we try to introduce you also to our pipeline. So the way we transfer those information and that knowledge is usually through the crystallization form. We have a crystallization form that we request you to fill in, giving us as much detail as possible so that we understand how your system works. And then we also tell you how things might or might not work with our pipeline. You also can get a library template. So if you want to ship us your compounds, we would want it in a certain format, and we need then the plate map for that library. So if you've never been to XChem, normally we would organize a 1-week training on site where you would go through the whole XChem process from the sample preparation to the data analysis. It's a small scale, basically, experiment of the fragment screening. And it gives you really a good overview of the pipeline. And for us, we can then start assessing the project and see whether we can identify major problems or not. So that training week is really 1 week on site. And then normally we have a characterized system that we know what to do on. You know, this type of experiments should be then for you a routine experiment. So you would then come back for 2 days on site to do the full scale fragment screening. And in terms of support, then we will be on site, but you would have in the lab some step-by-step documentation to help you. Then the data analysis support, this is probably the one that we struggle to cover properly during the training week. That's why we tend to have, again, some step-by-step documentation, again, some support by emails or conference call after you've done the full-scale screening. And we also initiated that XChem bulletin board that is very similar to the CCP4 bulletin board to allow you to ask questions, and whoever knows the answer then can reply. So of course, during the COVID time, we couldn't do all what we used to do, mostly because we couldn't get any users on site. So the way we handle mostly COVID projects right now is that we would ask you to basically double check the robustness of your crystal system in-house and on other MX beamlines if you need X-ray feedback. And then you would provide us the crystallization solutions and all the things that we need to crystallize your protein on site. Or you provide us the crystallization plates if you're local. And you would also have to do the data analysis on your own. Well, not on your own, but supported. What we will do then is the crystallization in the research complex. We will do the hands-on experiment here, and we would support you for the data analysis and the deposition. And there was quite a lot of work. So basically, Felicity moved a bit from the MX support group to help us in that sense on the logistic and the crystallization. So again, you would heavily rely on documentation online, but we also organize Zoom session for you, especially if you've never done anything with XChem. We are now starting to record all those videos so that you can go back to those training materials. And we're starting to also create drop-in sessions for data analysis. And this is more or less when we know that most of the users are on the same level of data analysis, then we can organize those drop-in sessions and you can basically fire in all the questions that you have for an hour, and we would be available to answer those questions. So what we did then between February and now, basically on COVID-19 targets, we had about 8 proposals going through. We've done 7 of them. 1 didn't work at all because the crystals didn't diffract at all. We've done more than 12,000 data sets on IO41. There are still 4 projects getting analyzed with 400 interesting hits. We've done 3 projects that are released into the public domain. And those are a bit the statistics that of those projects. And I think the most interesting is the success rate. So when we could do the experiment, we always had a hit between basically 3 and 7 or 8%, which is quite reassuring. Basically, we're pretty sure that we can do screening experiments. I think the tricky bits, and that's why we want to be sure you line up your medchem and computational chemist downstream, is that what you do with the hits is a much more complex story. So meanwhile, also what we've done in terms of libraries on the XChem platform during that time, we've been extensively testing the probing libraries. So those are libraries basically of fragments that are even smaller than 200 Daltons, and some of them are 3D-like. And those probing fragments are definitely not a starting point for lead compounds, but they can find hotspots in your on your protein for interactions. And this is really to help you guide a bit how you can then evolve your fragments. Then the other library we've been testing quite heavily is the EU Open Screen library. So this is a library that will be made available to the users when everything is in place on the EU Open Screen side. The nice thing about that, so it's 960 fragments. And the nice thing about this, I think, is that they can provide medchem consultancies for groups that do not have that expertise and would like to have those consultancies. So that's what they promise they will do. And so we needed to confirm that that library was valid in our hands, because it's at a lower concentration than what we normally do. So our libraries are usually at 500 millimolar, and that library is at 100 millimolar. But we found hits, and they are in sites that we are interested in some of the targets. So we're quite happy with that one as well. And finally, what we've restarted slowly since September is the user program, which you probably are more interested in. We had to pause everything in February, and so now we are just catching up on all the users that we had to cancel at that time. We've also tried to have one user on site, but then UK went back into lockdown. So I think this is going to be postponed for a while again. But it was a fully independent experienced XChem super user. And he came and everything went fine, very smoothly again. So far we've done 2 projects remotely and 1 on site. And so what you can get out of such an experiment, so those are the examples that are very recent from the work we've done on SARS. So the main protease, that was in collaboration with Martin Walsh and Neil Lonson's group. These basically, we did that screen within 3-4 weeks. And then the nice thing is that there was really a huge interest from the international community to suggest compounds to synthesize. And so that's the Moonshot program that was led by Alpha Lee, John Codera, Neil London, and Frank. And really that led to now some fragments are in the 600 nanomolar affinity, which is very, very fast from going from a fragment into a reasonable lead compound. Then the other 2 projects that are now released in the public domain is NSP3 macrodomain. That again, we did a very extensive screen of several libraries onto those targets. And we could really see a proper mapping of all the interesting sites and also potential merging and linking of the fragments. And while we were working on that, we were contacted by Fraser's group in UCSF, and he's linked to a ComChem group, Shoishet, which is a very brilliant group to be linked with. And they want then now to take forward all the hits that has been found on that target. And finally, on NSP13 helicase, that's the last one that got released to the public domain. Again, this is really the type of results you will see. You will find hits in all possible pockets that are available on the protein as well as, that's a drawback on the crystal packing. So you need to filter what is interesting and what is not interesting, but it definitely gives you a mapping of all potential interactions. And again, that one now is getting followed in the MedChem group in Oxford, and I think maybe by industrial partners. And of course, this is a huge group. So Frank sits at the Structural Genomics Consortium in Oxford and at Diamond. So this is the work involving the two groups, really. And this was the last photo we had just before the lockdown in March. And if you have any questions on the academic side of XChem, you can contact Frank, me, or Darren. And for the industrial side, you can contact Alex and Elsa, and all the informations are almost up to date on that link. And I think I'm on time. Thank you very much.